then I'm Jeremy Miller. I'm a scientist at the Allen Institute for Brain Science, and I will talk to you today about some of the Allen Brain Atlas RNA sequencing resources that we have. And feel free to interrupt me with questions or comments, or to tell me something didn't make sense. So, didn't transfer. Oh, there it goes. Okay. So I broke my talk into 4 parts. The first is I'll give you an overview of what RNA-seq actually is, and then I'll talk about the 3 resources that we have, which include RNA-seq data. So the Brain Span Atlas of the Developing Human Brain, the IV Glioblastoma Atlas Project, and then the Aging, Dementia, and Traumatic Brain Injury Study. So at the highest level, you can think of RNA sequencing as a microarray, but with a lot of additional features. Instead of looking at the intensity of a dot on a screen to try and understand, to get the amount of information for a gene, it's actually sequencing individual nucleotides. I'm not going to go through this to look through what all the differences are, but it's basically similar, but you get a lot more information. So the way that RNA sequencing works is that you first collect the tissue from wherever you're collecting it from. In our case, it's generally the brain. You extract the RNA, you then make cDNA and shatter the RNA shatter it into fragments, and then you sequence the fragment ends. So the output, is there a pointer, or can I use, you have one? So the output that you get out of it is basically a bunch of nucleotides of generally 50 base pairs long that are then aligned somewhere in the genome. And the way that you can quantify the amount of a gene that's expressed in a given sample is the number of those tags that end up aligned to that particular part in the genome. So what you get from that is, to measure the expression of genes, is basically, you basically get just the total number of these tags that you get within a given gene, and that would be the number that you get, which is comparable to the number that you get from a microarray. And this would be probably for another gene, you'd get something similar there. And using this kind of information, you can then compare gene expression to whatever phenotype you're interested in. So you could look for a correlation of expression of a gene against, age or some kind of a disease metric, or you could look at differential expression between 2 brain regions. Another thing you can do is you can choose any set of genes that you want, and then use those sets of genes to cluster samples in some way. So you could look for, say, a set of genes that are the most differentially expressed between, hippocampus and cortex. And if you get a sample and you don't know whether it's coming from hippocampus or from cortex, using that set of genes, you could potentially cluster it there. So another thing you get is information that you don't get typically from microarrays, is information at the exon level. So if you, instead of summing things up at the entire gene, you could look exon by exon and do basically any of the same kind of stuff I just talked about there. Or in addition to that, you could look for alternative splicing that may be potentially different between 2 conditions. So you could look for situations where, in one brain region you may get just exon A and B, and in another brain region you may get exon A and C. And even if the total level of expression of a gene may not have changed too much. So you get whole gene, individual exons, and you can also get a lot more complicated information that you don't get at all from microarrays. So this is what a genome browser typically looks like. If you map all of the reads for a given sample, you can get a cool visualization, first of all. You can also get differential expression, like without paying any attention to what the gene is. You could look for whether, how many reads just pile up in this area. You can also look for novel exons. Maybe there's an exon here that hasn't been annotated, but you get a lot of reads in that area. In the case of cancer, you could do fusion gene detection. You could look for single nucleotide variants, a lot of things along those lines. Most of this stuff is beyond what I'm going to talk about, but it's things that you can do if you want to download the raw data and take a look at that. So, any questions about what RNA-seq is?
So, and I'm learning this. So you don't read the entire fragment, you just read the ends of it.
You just read the ends of the fragment.
So how can you tell if there's a mismatch? Because
if the mismatch, so this is mapping against a known genome or transcriptome, and if one of the nucleotides in the end that you map doesn't match with the other 49 within that read, then you'd call it a mismatch against the genome.
Okay.
So,
but every nucleotide is probably going to be covered in that.
No. So if you couldn't do this for like genome-wide type of re-annotation.
Inside the gene, every
Probably not. I mean, it depends on how much. This is a gene that has a lot of expression. Typically, you'll get stuff like this where there'll be a number of genes that pile up. There'll be some spaces in there where you don't get any information. If you just get one, you wouldn't necessarily believe it because there's some amount of error that gets thrown in there. But if you get a giant group of them like this, and you find 15 or 20 of them in a row that all have the same mismatch, you can be pretty confident that that actually is a mismatch. So that's one of the sort of least common uses for RNA-seq, but you can do it.
And one other question. So I hear people talk about million reads. Like what's...
So the number of, when you break the RNA, the cDNA here, these fragments, every time you grab one of those, that's considered a read. And the more of them, it's a stochastic process. So if you were to take 1/10 the number of them that you end up with, you'll get roughly the same distribution that you had before, just 10 times lower. And so a lot of the genes that you have really low expression of, you would need to get more reads or sequence deeper to be more confident in that call that you get.
In what state the tissue was before you extracted the RNA?
What state is the tissue in?
Yeah.
So the way that I think we usually do it is you freeze it, section it, and then cut out a chunk of tissue, and then I'm not exactly sure what the experimental process is, but it's very similar to what's done with the microarray. What we're doing now is on single cells, so you can actually get individual cells and run RNA sequencing on individual cells. So pretty much, I think any kind of tissue you can do it with. I don't know if you can start with fixed tissue and get any kind of good information out of it. Okay, and so this is basically showing the same thing, but just the data files you get out. So the raw data comes out in what's called a FASTQ file, and this is just a list of all of these reads before you actually map them anywhere. So you'll just be probably a couple million strings of 50 base pairs or sets of 50 base pairs. And the next step is when you map them to a specific genome or transcriptome, you get what's called a BAM file. And this is basically exactly what these are. It's this pile up that you get right here of all of the reads and where they line up along the particular genome and transcriptome that you used. What we do for our releases is we anonymize them, where we don't actually have the information that would allow you to decide whether there was a variant change here, but everything else you can keep. You get the positions along the genome of all the information, you just don't know the exact nucleotides, and this is to keep the donors anonymous that we provide. And then there's a thing called a bigwig file, which basically gives you this coverage plot, which often is what people that is all that you need. And then the normalized data would be compressing all of this stuff down to one number per gene or one number per exon, and that's what would be comparable to what you'd get for microarrays. And what, if you see any of the heat maps that I'll be showing from the website, that's the number that goes into those.
So you align to a specific genome. So where do you get that? Like where do people get the reference genomes?
The reference genomes are, I think you can get most of them, they're very standard. You can get them from, I think, what is it? I'm completely blanking on the university that has most of them, but there's like an HG38 is the most recent one for human, and that's the one that we're using here. The transcriptomes are a little bit less standard. I mean, there are a few of them. We're using RefSeq, which is the most conservative of them, but that will miss a lot of things that people aren't entirely sure are actually genes or transcripts, but some people say they are and some people say they aren't. And I mean, we use the most conservative one where everything in there people are pretty darn confident is it actually a gene. And I think at least for one of our data sets, we have the ones we used as a downloadable file from the website. Okay, so the first one I'm going to talk about is the BrainSpan Atlas of the Developing Human Brain. We have a few atlases that are similar. This BrainSpan one that I'll talk a little bit about, in detail here, is basically coarse coverage. So you take a big chunk of tissue, and that's where the RNA comes from. But it spans a large amount of time, from very early prenatally up until like donors in their 40s. And so this is high temporal coverage, relatively low anatomical resolution and coverage, and it's done using RNA-seq. We have another atlas from BrainSpan that I won't be talking about that is using microarrays prenatally and has a lot more finer anatomical resolution, but only 2 time points over 4 brains. And then there's another non-human primate atlas that's kind of somewhere between these two, which has 10 different time points, anatomical resolution as good as this one, for a few brain regions, and is also done using microarray. So if you're interested in development, these would be the 3 atlases that you'd want to look at. This is the one I'll talk about that uses RNA-seq. Okay, and so here's kind of more details of all the brains that we have in this atlas. So there's several of them that start pretty young prenatally. I think there's about 30 or 40 of them in total that were done using RNA-seq. For each of the brains that we have across the time point, there's 16 brain regions that were collected. 11 of them are in neocortex, and then there's hippocampus, amygdala, striatum, thalamus, and cerebellum. And let's see, and this was done using RNA-seq as I mentioned. There are several other components that were also collected on many of these same donors across time. There's also, epigenomics, there's in situ hybridization, there's several reference atlases. We have some methylation data, histone data. I don't think we actually have on our website single-cell RNA-seq, but there's a lot of other data that's also available that you could tie into this RNA-seq data if you're interested in doing that. Okay, so some of the broad results that we get from this atlas are probably what you'd expect. This is a principal component analysis showing sort of the highest amount of variation in the data. The first principal component, which explains the most variance, is how genes change with time. So there are a lot of genes that are either very high or low expression prenatally, and then change drastically over the course of development. If you look within a time point, so this is one of the prenatal time points. Here's another one, and here's an adult time point. These are color-coded by brain region. So within a given time, you find basically that the biggest source of variation is changes within brain region. So these are almost a positive control to make sure that our data is telling us what we would really want it to tell us. Okay, and with this atlas, we don't have a resource paper about it, but there have been several papers that have been published using these data. So if you want to get a sense of how other people have used these data, I have a few of them listed here, but you can just do a search for BrainSpan in PubMed or something, and there's several other papers that have used these data for in their work. Okay, so this and all of the other tools from the Allen Institute are freely available to download and use however you want. This one you can get either by going to the main Allen Institute website or by going to brainspan.org. This is what the main page looks like, the home page looks like. And if you want to look at the gene expression data on the gene level analyses, you'd go to this developmental transcriptome link, and you can use either the gene search or the differential search, or you can click on any of these gene lists that we've. Do you know where these gene lists came from, Terry?
Online search tools.
Online search tools.
Yeah, there.
Okay. And so the gene search, you just type in the gene, and it'll give you back all the data for that gene. I'll show you an example of that on the next slide. You can also do a differential search where you can put in any subset of donors, of structures or stages, and compare it against any other subset of structures or stages. So in the previous slide when I showed this principal component analysis that showed differences between either region or between time, you could look for those genes that make up those strongest differences yourself by doing this kind of a differential search. Then once you do the search, you come up with a page that looks something like this, that will have one or potentially many more genes that are listed here. And you can sort these genes either by the stage, so by the age that it's in, or alternatively by the brain region, by hitting this little arrow in the corner to reverse the search. In this case, this is a gene that marks excitatory neurons, and it has high expression pretty much everywhere. I actually said highest expression in thalamus and striatum. That's supposed to say lowest expression in thalamus and striatum, but it's expressed in most other places and increases with time. So there's more red over here at the later stages than at the earlier stages. Now, if you have a gene that you are interested in and you want to find other genes that have similar expression patterns, you can do that by clicking on this Find Correlates button over there, and you'll get back some kind of a search like this, where there's a set of other genes that have similar expression patterns to the one of interest, and they're ranked by how well correlated they are with your gene of interest. So, but since this we know is a marker for excitatory neurons, potentially many of these other genes are also markers for excitatory neurons, and that might be a good way of starting if that was the question that you were interested in looking at. Okay, so another thing that you can do with these data that you can't do generally with the other microarray atlases we have is to look at the exon level. So this is showing the overall expression of the entire gene across all of these samples. If you click on the gene symbol for your gene of interest, it will bring up the same plot, but will split out your gene exon by exon. So this is looking at all the different exons for this SLC 17A7. In this case, most of them look pretty similar, except for these last couple don't really have very much expression. And we have both RNA-seq data as well as exon array data for many of the same, most of the same donors that are in this data set. So here you could get sort of two different kinds of information about the same gene across the entire data set. And these data, as well as I think almost any link you're at for the heat map, you can download the data by clicking on the download the data link. The other way of downloading the data, which I'll talk about shortly, is downloading all of the data from the download page. So this would give you basically everything you see here. Are there any questions so far about any of this?
So when you download it, it's in the normalized to the exon?
I believe when you download the data, it's normalized to the exon in this case.
Okay.
So so you'd have some sort of a distribution across the entire gene here, and it would just take whichever ones of those reads fell within this exon would be the number you'd get here. I think this is also scaled so that red's the same for all of them, even if there's more of them. Actually, that's not true because these are all lower, so they're all scaled collectively, so that it's all scaled together. Okay, so then if you want to download all the data, or any components of the data, you go to the download link from the main page. You can get the whole data set summarized either to the genes or to the exons. I think these would be the numbers. Maybe a scaled version of these numbers is what you get when you download any of the pages individually. And so you can get all the data here. Here's sort of an outdated version of it. You can get data from a bunch of the other components of BrainSpan, which I removed just for this slide. These bigwig files would be the way that you would view just the pileup plot. The advantage of these is they're much smaller and easier to deal with than this MRF file, which is all of the reads in order. This is a slightly different format from the anonymized BAM files, but if you go to this paper, which you can look up with RSeq tools, there's a way of converting them directly into the anonymized BAM files, and you can get all the information that I talked about before for each of the samples. And then there's other things that are available for BrainSpan. There's the prenatal laser microdissection microarray dataset that I mentioned earlier. There's a lot of in situ hybridization. There's reference atlases, 2 for prenatal and 1 for a postnatal time point. So you can look those up. There's also documentation and help for this and any of the other atlases if anything is unclear. And then another way of accessing the data, other than the download page or the website, is through the API. And so here's brain-map.org/API would be the way you'd get to that. And this gives you more programmatic way for getting at the data for a lot of the different atlases. So you just click on whichever atlas that you're interested in, and it'll take you to all the information that's available for the API on this atlas. I'm going to refer you to Terry for information about the API because I don't really use this, but it's a very good help for all of them. And as part of the help, it tells you what's actually available as part of the API. And there's further down on the screen, if you were to scroll down, there's examples of code that you can type in to get the particular information that you would be interested in getting using this resource. Okay, so are there any questions about this atlas at all? Okay, so then the next atlas that we have that includes RNA-seq data is the IVGAP, or the glioblastoma atlas. This atlas was a partnership between the Allen Institute, the Ivy Foundation, and then the Ivy Center for Advanced Brain Tumor Treatment at a Swedish hospital. It's a large public resource that has 50 patients, 500 tissue blocks, 270 transcriptomes, which is what I will focus primarily on, and a lot of machine annotated pathology images, and a lot of in situ hybridization. All the data is available either at our website or at the IVGAP website, and it was released last year. The goals of this project were to identify genetic signatures of cancer stem cell niches and anatomic structures, and to measure intra and intertumor heterogeneity, and to identify correlations across data by modalities that predict patient outcome. And yeah, there are several other collaborators that were also involved in this project from the beginning.
Has this been published a great deal?
So we're working on a paper to describe it. I don't know if anyone's published on it at all.
I'm not sure. We should actually do a search just to see.
Yeah, I don't know.
He's got me the big product. He didn't want me to work on it.
All right, so there's two basic components to this, two data types that were primarily used in this data set. There's in situ hybridization that spans 6 different projects, but overall it's about 500 different genes on 42 patients have in situ hybridization data of some kind in this resource. And you don't have to know the differences between these projects to be able to find whatever data is available on whatever your gene of interest is in the website. You can just type in your gene and it'll bring up whatever's there. The other component is this laser microdissection and RNA sequencing, where basically you cut out individual anatomic structures or stem cell niches and tissue sections and then sequence it to look for the entire transcriptome using RNA-seq, as I talked about earlier. And this is what I'll talk about mostly. And so this RNA sequencing part data was split into 2 different parts. The first part was on anatomical structures, and when I'm saying anatomical structures, what I mean are basically different kinds of cells in the tumor based on histology. So the leading edge is primarily it's primarily normal cells, but there's a handful of tumor cells in there that some people think maybe the tumor cells that sort of go out into the tissue and start causing problems in there, which is why this area is of interest. What we used it for primarily is a control, because with RNA sequencing, it's 97% normal tissue, and the biggest signature in there is normal tissue. But potentially there could be genes in there that are marking those handful of tumor cells that could be of interest to look at. Infiltrating tumor is basically an intermediate zone of the tumor where it's 10 to 20 tumor cells per 100 normal cells. The main sort of part of the tumor is the cellular tumor, which is almost entirely tumor cells. We also use this as a control for a lot of the data in both parts of the RNA sequencing data sets. There's also MVP, which are blood vessels in the tumor core, and then there's this I'm not even going to try and pronounce it even after working with this data set. PAN, which is basically this wall of cells that surround necrosis at the core of the tumor. And so these were all collected separately in 3 different blocks across 8 different tumors, with the goal of looking at differences between these regions as well as differences within tumor versus differences between tumor, to try and get an idea of inter versus intra-tumor heterogeneity. Okay. All right, and so this is what some of the data looks like. Here are some examples of genes that are very, very specific to these different regions. It turned out that infiltrating tumor looked basically halfway between cellular tumor and leading edge, so we don't have anything about that here, but there are several genes that are, here this one's 3-fold more highly expressed in here in leading edge than in infiltrating tumor, and is basically absent in all the other cell types. There are other examples in the other brain regions that are very specific to, tumor regions that are very specific to these structures. If you look at multidimensional scaling, you get a very good separation between these different types of structures. So this is MVP, this is the leading edge, and then this is, I think, PAN. So it separates very well by gene expression. They also separate by histology, as I talked about, to the point where the histology was there was actually an automated system that was used to determine the different brain regions that was very accurate. And that was used for how the tissue was laser microdissected to go into the RNA-seq in the first place. And then there's in situ hybridization. For a lot of these same genes are very specific to particular regions. So we have, if you want to look at these different anatomic structures, you can do it by gene expression or histology or in situ hybridization. We haven't really looked into the sort of base pair level analysis in a lot of detail, but potentially these are some very strong differences. There are probably going to be a lot of other differences at sort of individual base pair level that you could get at if you wanted to using the RNA sequencing data.
Is the histology annotation code available?
I don't know. I should know that, but I don't know. I know that. Yeah, I don't know. Do you know?
Write that down, so I can get back to you on that.
It's a very useful tool.
Yeah, I know for some of this annotation, I don't know if what was used here. It's a program that's commercially available, but I'm not sure whether that's the same as what is shown here. So yeah, Terry, I'll have to get back to you about that. Okay, and then the rest of the RNA-seq data was on these cancer stem cell niches. So there are known sets of there's I think 17 genes were chosen here through some level of knowledge in the field and a pre-screening of in situ hybridization that are basically stain parts of the tumor in ways that you would not be able to look any different through histology. And the hypothesis is that these are stem cell niches, similar to the kind of stem cell niches that you get in the brain, where new tumor cells would be developed here rather than anywhere else. It's a hypothesis. It's not really clear whether that's actually what's happening, but that was the way that we went about looking for the areas to sequence for this data set. Basically look for areas of the tumor that were stained by a particular gene of interest, cut those pieces out, and then in the same tumor cut out a piece of cellular tumor where none of the genes that we were looking at were staining. So it was basically, this is kind of what it looks like. There's 34 different tumors. For each tumor you take a piece of cellular tumor as well as I think we picked up to 4 genes that had this kind of patterning in the same tumor. And in total there are 148 tissue samples across this part of the data set. And again, you don't have to necessarily know which data you're looking at. It's all part of the ontology includes, what region this came from and whether it was part of this or the other project. Okay. And so here's a couple of examples of what it looks like. I think this one actually stains most of pan, but not all of it. This one labels a perivascular niche. I don't have examples of it here, but there are some where you just have cellular tumor. It looks pretty flat, and it's just in some spot in the middle that you can't tell by eye is any different. Okay. So here, whoops, oh, here it goes. Here's an example of what we've done looking at these data. This is a very complicated plot, but I'll try and go through it. Because the different brain regions, or the different structures that we looked at are so, you know what, actually I'm not gonna go through all of this. It's probably too much information. If you care about this, I can talk about it later. But basically the cancer stem cell niches that we found fell into pretty much 3 categories. Some of them were proliferating cellular tumors, some of them seem to be focused around blood vessels, and some of them seem to be related to hypoxia and immune response, and you can get that information from RNA sequencing. And I can tell you about this later if you are interested. So here's what the data set looks like. You can get to it through glioblastoma.alleninstitute.org or also through the main webpage. There's the home page. Just like the BrainSpan Atlas, you can do the same kind of gene level analysis, either with a gene search or with a differential search. Here you can also click on these links down below that will give you the top genes that are specifically expressed in the anatomic structures. One different feature here is you can actually select any of the tumors that you want for either your gene search or your differential search by going through and sorting them either by the age of the donor, by their gender, by the tumor name, by the molecular subtypes that were present in that tumor. And so you can do a search either for all of the tumor, all of the data, or just any subset of it that you're interested in. And then again, this will bring you back to a similar looking heat map from the BrainSpan data set. You have many of the same features. One different feature here is that you can click on this tumor subblock, and if you want to actually take a look at whatever tumor, in this case has really high expression of MET, it turns out there's a subtype of cancer tumors that has high expression of this particular gene, and you could go look at that block and see how that's different than the other ones if you wanted to by clicking on this. Otherwise, many of the features here are the same as from the other atlas. Let's see. Okay, and here you can download the data, again from the download site. You can get any of the sample information, gene level data, and then the anonymized BAM files, and other information about the tumors available here. And for this resource, there's a lot of other information available about the tumors. I think there's MRI on the patients with the tumors in their head that you can get through the IV GAP resource if you want to.
I was wondering, what kinds of sequences do they have?
I think there's genome sequencing on all.
Sorry, MRI sequences?
Oh, MRI. I don't know what kind of MRI they have there, but we can get it. Yeah.
Yeah, there's diffusion there.
Yeah, I'm not sure. But yeah, I could look it up.
DTI?
Yeah. Are these taken from their clinical studies?
Yeah.
I think it's the clinical, yeah.
There might be some diffusion, but most likely it's T1
Well, how do you go to look?
Yeah, sure.
But I think those you actually have to contact them to get access to. I don't think you could just download them. But we can look it up.
I have a password to get in.
Okay, you can look it up. All right, and there's no exon level visualization in this resource, right? I don't know of one.
No.
Okay, I don't think so.
Yeah.
The API here is a little bit more extensive based on the number of icons. So there's more things you can do through the API here for both the RNA-seq and the ISH. And if you want to do that, you can go to the help, which is very good. And here's the website for doing that. Or you can go to the API same way as before and click on the glioblastoma link. Okay, and then the last data set that I'm going to talk about for RNA sequencing that is not showing up on the screen. Hold on.
Did it move on the computer?
Mm-hmm.
has to go all the way to the satellite and back.
Okay, yeah, it made it. Must have been hit a delay. Is the aging, dementia, and TBI study, or traumatic brain injury study. And so this was originally designed as a study of the long-term effects of mild to moderate traumatic brain injury. We went in not expecting to find much, so also designed it so we could look at aging and dementia as well. And so the reason for looking at this is that severe traumatic brain injury contributes to about 30% of all the injury-related deaths in the U.S. The long-term effects of repeated mild head injuries is widely reported. So if you've heard anything in the news about the football players and boxers, that's different from what we're looking at. That's a lot of different repeated head injuries over the course of a long time. But if you fall off your bike and knock yourself out once when you're a kid, whether that has a long-term effect, that sort of a question is what we're looking at here. And that hasn't been very well studied. Also, we wanted to look at aging and dementia, since dementia affects a large percentage of the aging population, about 14% of the population over age 65, and the incidence roughly doubles every 5 years. And the majority of these cases are diagnosed as Alzheimer's disease. And so there's no cure for Alzheimer's disease, despite there being a lot of known pathologies associated with despite the known 100 years of study. And there's two main pathologies associated with Alzheimer's disease, amyloid beta and tau. These are very widespread in really old individuals and don't really do a very good job of correlating with Alzheimer's disease anymore at that point. There's also evidence that these, as well as Lewy bodies, in really severe traumatic brain injury can show up. So we can use the same pathologies when we're trying to study traumatic brain injuries when we're trying to study aging and dementia. And so one question that potentially could be answered using this resource is what makes certain people less prone to some of these deficits of aging than others. So this study was designed, was a subset of the Adult Changes in Thought study, which is a Seattle-based study it's basically random enrollment of cognitively normal group health patients that are age 65 and older. So anyone who's enrolled in group health and makes it to age 65 without any kind of cognitive deficits has the option, a subset of them have the option of being part of this study. So as of now, there's about 1,000 participants in this study. Sorry, about 5,000 participants in this study, of which about 1,000 of them are diagnosed with dementia. A subset of those people agree to donate their brain when they die, and about 650 of them have done that so far. And what we have is 107 brains from this 650 cohort. 55 of them are people who had self-reported 1 to 3 lifetime traumatic brain injuries with loss of consciousness. And then there was a cohort, a non-exposure cohort of the same number of donors where they didn't report traumatic brain injury and they were matched by age, sex, post-mortem interval, and year of death. And then we had to exclude 3 for low tissue quality. So this is designed as an exposure, non-exposure cohort. We have a table that you can download, which will allow you to correct this correct for biases introduced by this case controls type of cohort, to look at the entire cohort as a whole, if you wanted to do that. There's also a white paper about how to do that. But the basic idea was you take a relatively small subset of this cohort, you characterize the brains really well, and then we'll obviously provide all this available online. There's a lot of different kinds of information that are available. There's the RNA-seq, which is in this one little spot right here, but there's also in situ hybridization for genes marking cell types. There's some different kinds of markers for the pathologies that I was talking about, the tau and amyloid and alpha-synuclein pathologies, as well as inflammation, microglia, and other things. And all of this stuff is available as for all of the donors and brain regions that we have in this resource. And so we have temporal, parietal, cortex, gray matter, parietal cortex, white matter, and hippocampus in this resource. So overall, there's about, I think, 370 samples for the 4 brain regions across these 107 donors, and you have all this other information also. Okay, so does the setup of this study make sense? Okay, so then one thing we have for this resource that we don't have for most of the other ones is we've included on the website some of our initial looks into what you can do with the data and what kind of results you can get from this data set. It's under the Overview tab. We call them data snapshots, and so this allows us to explore some interesting RNA-seq findings. Sort of the first one that I'll talk about is this gender. There's a set of genes that are known to be expressed in males as females. These are the ones that are on the X and Y chromosomes predominantly. Here's about 25 of them that are expressed almost entirely in males because they're on the Y chromosome. Here's one that's expressed entirely in females, basically is on the X chromosome. What we can do is we can take all the samples from each donor and cluster them based on these genes, and you'd expect them to group into males versus females. And that's what happens. You can, using that set of genes, we use TSNE, T-stochastic neighborhood embedding, which you can think of it as basically a way of grouping together samples that have similar expression patterns and separating ones that have different expression patterns. We can talk about exactly what the details are later. So this does separate females from males. Along with these data snapshots, we have parallel coordinates on the bottom, which allow you to highlight certain subsets of the donors that you're interested in, or the samples that you're interested in. So if you're only interested in temporal cortex, you could just highlight this and all the other ones would turn clear. You can also recolor based on other metrics. So if you're interested in, well, within males and females, how do people based on these genes divide based on this tau? You could do that too. You could color code by the tau. And there's several other of these data snapshots that I'll also go through. But that's the basic idea. You get you pick a gene set, you then cluster a set of samples based on those genes, and you can color code them or subset them based on any of these other metrics that you'd be interested in. Mm-hmm.
So one thing that I found really interesting about this data set is that the data that you can actually download from the website is now organized, I presume, in the way that it was to create these things. So it's extremely neatly organized in this extremely tidy set of tables that you can very readily read into your computer memory and start analyzing. So I thought that was phenomenal for this data set. It's really start doing just exploratory data analysis with these data.
That's good. That's the goal. So I'm glad it comes across that way.
Because it is high dimensions.
Yeah, it's a complicated set of stuff, but they're linked. There are several tables. You need several tables because you can't put it all in one table. But those are linked through keys that make it really easy to go from one to the other.
Oh, that's cool.
Start exploring the data.
Good.
That was great. So you don't need to know anything about the API and backing this, and you don't need to know anything about. Actually, you don't need to know a lot about the semantics of the data itself. It comes in this table. You can find the linking variables between the tables. You can say, oh, there's this variable here and this variable there. And I think people who don't do biology, but do say machine learning, that would be a data set I would point them immediately to and say, okay, yeah, find something in there.
Oh, that's good to know. Okay.
Yeah, we'll definitely pass it on. Easy access to the data from this data set. It'll be good for the people who put this together to hear. Here's another example where we took the top genes that were differentially expressed between the different brain regions, and then they separate out by brain region. So here's hippocampus, here's cortex. The cortical white matter samples generally split out from the cortical gray matter samples. So
just to understand the preference, you assess every gene separately and see how well they separate between the four brain regions, and you take the top, how many you have here, and then you do TSNE on it?
Yes. So in this case, we did just a differential search in the website for the parietal and temporal cortex samples versus the white matter hippocampus samples, and just took the top 15 that were highest in each, and then used those 30 genes as the basis for this TSNE plot.
Okay. So that's kind of self-fulfilling.
It is. It's entirely self-fulfilling. However, if you do that with the TBI, yes versus no, take the top 15 that were highest in TBI exposure versus non-exposure, the top 15 in non-exposure versus exposure, there's no separation whatsoever. So it's not guaranteed if you have a negative signal that you won't necessarily get a separation here. So if there is a signal, then this will, this is what you'd expect to get. We were not at all surprised to find this. This was supposed to be kind of a positive control. But the fact that we did exactly the same process and didn't find anything for traumatic brain injury was a way that we could, one of the ways that we looked at to find that, well, there's probably not a signal between these two different groups.
So how many genes are there? 20,000?
20,000, or I think it's more like 50,000 transcripts, I think. I mean, so we have several other lines of evidence that we've looked at and we're unable to find any difference between the TBI exposure and non-exposure. You're welcome to try and prove us wrong if you want. The data is freely available here.
But that's like a thought experiment where we generate 20,000 vectors for 100,000. So for 100 subjects, and it's completely random. It's generated from a normal distribution.
Yeah.
And now we're gonna, out of those 20,000, okay, and then we generate another vector, yes or no, for each of the subjects, also random. Because there are so many variables, 20,000 variables, there's a non-trivial chance that 10 of them will be separating this completely random vector, yes or no. And the more variables you take into account, those 40,000, finding 10 that separate would be even easier.
Yes.
Just by chance.
Yes.
So the fact that for some you haven't found and for some you did find, this is not, I don't think that...
So what I will say is this is a good visualization. This is not the specific statistical test that I'm using as the basis of saying there's no difference. It's a good confirmation of all the other statistical tests that we've done where we didn't find any difference here. Brain region, we better find differences because we have several other atlases that show a lot of differences between brain region. And we've done, comparisons. I see your point, and we've definitely taken that into consideration. I mean, the multiple test correction for this data set is a nightmare.
Because it's cheaper to test 10,000 more genes than 50 more brains.
Yeah. I mean, and here we have multiple, I don't know if I can show you, but there's multiple different ways of measuring the tau and the amyloid beta that means slightly different things, and sometimes they'll give you borderline results, and the question is, which one do you believe? Do you believe the positive result or the negative result? So there's a lot of that kind of thing that definitely has to be taken into consideration when looking at these data. So that's definitely a point well taken.
So that's interesting, because with tau and amyloid beta, we have this very unique data set in one sample where they propose a new radioligand for PET studies that is supposed to diagnose people exposed to severe concussions over time. We're talking about football players, etc. In vivo, which is apparently very hard. I'm not an expert in this, but usually that is done post-mortem.
Yeah.
And it would be interesting to compare that distribution, because we have a whole brain PET map for it.
Yeah.
With the candidate genes here.
Yeah. The first step would be finding candidate genes here, and what we're arguing is that there really aren't any. That doesn't mean there aren't any, we just haven't been able to find them in our search. This also is not the same kind of data. This is looking at individual traumatic brain injury, but there are, I think, several papers that have been published looking at that kind of gene expression data set. I don't know if the data is available, but you could look that up. So. All right, so we didn't really find much related to dementia, which was a surprise. I think I'm going to skip over the results here, but I can talk about them. Basically, I can talk about them after if anyone's interested. Basically, we found a set of genes that were correlated with tau, and that agreed with data from a previous data set. And the biggest signature we found is that there are a lot of genes that are really related to RNA quality. So the tissue or the donors that had high RNA had high RNA quality, there are a lot of genes that were differentially expressed between those and the donors with low RNA quality, regardless of what any of the data in the tables said about their dementia status, TBI status, tau, any of that other stuff. And that was actually tied in a little bit with dementia status, because the donors with dementia tended to have lower RNA quality. So disentangling which gene expression changes were related to dementia versus RNA quality, I think, is something that should be taken into account when using these data.
Did all the dementia patients have low RNA quality?
No, so that's what this plot is. It's very highly overlapping, but they're slightly lower with in dementia than control as a whole. I'm sorry?
What affects the RNA quality?
So that's a whole nother discussion. We measured the RNA quality using the RNA integrity number. It's a measurement of kind of how degraded the RNA is. The agonal state, like what situation the patient is, prior to death, is a good indicator. A lot of it's just not really well understood. But yeah, but we have a measure for it that's very reliable for whatever the reason is.
I think they need the room.
They need the room?
Clock.
Okay.
Is there something else happening at 4 o'clock? Do you know?
Yeah, there's another talk.
Okay.
Is it happening in here?
Yeah.
Okay.
Let me skip to everything except to just mention that we do have a genome browser that goes along with this this data set that you can access by clicking on, where is it? Well, there's a genome browser button you can access it with. Most of the other features of this are the same, and I can show you anyone individually if you're interested in using this resource. Okay.
Okay. We can head out to that. Thank you.
Okay.
And then